Artificial insemination. In the first part of artificial insemination, we had already briefed about semen collection methods. Following this, let's talk through some aspects of semen analysis in this presentation. Semen evaluation. Semen preservation. Estrus detection and ovulation. Insemination methods. Advantages and disadvantages of artificial insemination. Semen evaluation. The semen is analyzed for various factors. The first factor is volume. The ejaculate volume is measured by collecting the sample directly into a graduated vial. Possible reasons for decline in semen volume include use of young bulls, frequent ejaculation or incomplete or failure of ejaculation, method of collection. The volume of bull semen varies between ejaculates, individual bulls, breed, and age. Semen volume for Bos Taurus bulls in Brazil was reported to be 6.9 milliliters and 8.2 milliliters in different years. A few of the common normal values for different animals are listed in this table. Semen volume varies from 2 to 10 milliliters per ejaculation for bull, 30 to 250 milliliters for stallion, 5 to 1.5 milliliters for ram, 150 to 500 milliliters for boar, and 1 to 25 milliliters for dog. The second factor is gross appearance of the ejaculate. The ejaculate is typically clear, milky white, or cloudy gray, with a jelly-like texture. Change in color implies an abnormality. If the ejaculate is dark red to pink, it indicates hemorrhage in the male reproductive tract. Yellow color is due to presence of urine, and curdy appearance detects presence of infection. The third factor is mass activity. A drop of semen is placed onto a slide without cover slip, and the mass activity is evaluated under low-power microscope. The good quality of semen is evinced by a rapid wave motion with formation of eddies at the end of waves. The fourth factor is sperm concentration. It refers to the number of sperms per ml of semen. Prior to sperm count, semen is added to a diluent containing a small amount of formaldehyde to kill the sperms and prevent the sperm movement. Total sperm count, abbreviated as TSC, is the total number of sperms in the entire ejaculate. Total sperm count is expressed as the product of ejaculate volume and sperm concentration per ml of semen. The two common techniques to determine the sperm concentration are hemocytometer and spectrophotometer. The fifth factor is sperm motility. It is defined as the percentage of sperms that are progressively motile. A progressively motile sperm swims rapidly forward in a straight line rather than moving in circles, as shown in this figure. The factors that affect sperm motility are exposure to heat and cold, wrong pH, sexual inactivity. The sixth factor is sperm morphology. The morphological abnormalities of a sperm can have an adverse impact upon fertilization, and also embryonic development. Fertile bulls show about 90% of the morphologically normal sperms. Some common sperm morphology abnormalities are represented in the images here. Bent tail or midpiece, double-headed sperm, coiled tail, detached head, elongated head. Semen preservation. The primary goal of semen preservation is to maintain the fertilizing potential of spermatozoa over a long period of time in a pathogen-free environment. The number of sperms per semen dose differs between species, and depends on the type of preservation method, that is liquid or frozen. The freshly ejaculated spermatozoa are activated by the seminal plasma ingredients. Usually, semen extender is added to the semen, to increase the efficiency of male gamete, to dilute toxic elements in seminal plasma, and to provide nutrients for the spermatozoa. After the addition of semen extenders, the semen is frozen down in multiple plastic tubes, known as straws. The straws are then stored in liquid nitrogen at -196 degrees Celsius, until they are needed for insemination. The conventional semen extenders include milk or egg yolk, that protects against cold shock, antibiotics to protect against contamination, buffers to protect against pH changes, glucose provides energy source for the sperm, and glycerol acts as a cryoprotectant. Fresh semen is used immediately or after collection. Examples: turkeys, human beings. Stored semen is used after storage at reduced temperature. Examples: horses, pigs, dogs. Cryopreservation is currently the best technique for achieving optimal freezing and thawing rates for a specific specimen. Glycerol and DMSO are commonly used cryoprotectants. Two methods of freezing and storing semen. One is liquid nitrogen at -320°F, and the other one is dry ice and alcohol at -100°F. Example: bulls. Liquid semen preservation. Semen is preserved in the liquid state at temperatures above 0°C, and is then stored in glass ampoules. Merits are low cost, and avoids chilling injury. Demerits are limited lifespan, that is 1 to 4 days, and high risk of bacterial growth. Estrus detection and ovulation. Estrus is the phase where the female is sexually receptive to males. And it is also referred to as the heat phase of the female. Estrus detection is crucial if the female is to be inseminated at the correct time. Electronic monitoring systems are used for continuous monitoring for estrus activity twice daily, at an interval of 8 to 12 hours to proceed for insemination. A couple of behavioral and physical signs of estrus include edema and hyperemia of the vulva, vocalization, mounting other cows, vaginal mucus, restlessness. Timing of insemination is crucial to obtain good results. Insemination should be carried out on the same day, if cow shows estrus in the morning. If cow shows estrus in the afternoon, insemination should be done on the next day morning or early afternoon. Insemination methods. There are different methods of insemination, such as rectovaginal method, speculum method, vaginal method. In this method, the speculum is placed in the vagina of the cow. The inseminating tube is passed through the speculum, and the semen is deposited at the cervix. In vaginal method, the inseminating tube is positioned at the insemination site, with the hand passing through the vagina, and the semen is deposited. There is slight risk of contamination and injury of the female genitalia in this method. Rectovaginal method. The procedural sequence in this method is pictured here. To begin with, insert the breeding gun into the vagina along the left gloved hand. Allow manure to pass over the top of the hand. Grasp the cervix, and push it forward to straighten the vaginal folds. Later, the semen is deposited into the uterine body with a plunger, and finally, contraction transports the spermatozoa to horns and oviducts. Let's talk over a few advantages of artificial insemination. Artificial insemination prevents the spread of infectious diseases that can be passed on when animals are in close contact or share the same environment. Reduces both genital and non-genital diseases in the farm stock. Prevents sterility by eliminating many genital diseases. Higher conception rates. Swiftly passes the desirable traits to many offspring. Maintains genetic diversity. Breeding can occur in the event of physical, physiological, or behavioral abnormalities. Conservation of rare breeds or endangered species. Least invasive and extremely cost-efficient. Disadvantages of artificial insemination. Some males shed virus in semen without clinical signs of disease. Some bacterial pathogens are resistant to antibiotics in semen extenders or can avoid their effects by forming biofilms. Requires well-trained operations and special equipment. Decline in fertility in dairy cattle and horses, associated with an increase in artificial insemination. Loss of genetic variation. Requires more time than natural services. Low fertility due to improper cleaning of instruments and insanitary conditions. If the bull is not properly tested, the spreading of the genital diseases will be increased. Thank you for watching. Please do like and share this video. Also, click on the subscribe button and hit on the bell icon to get the notification of our latest videos.